Hello future doctors, how are you all? Welcome back to MedSteps YouTube channel. So now we are following first aid guide musculoskeletal system physiology. So in this video mostly half of the physiology we are going to discuss. Okay, so main thing is our topics in this video will be organization of a muscle cell, types of skeletal muscle fibers like type 1 and type 2, and muscle proprioceptors and Golgi tendon organ and sarcomere and then neuron action potential and absolute refractory period. So these are every topics we are going to cover in this video. So without late, let's get started. So first of all, in skeletal muscle we have to know the basic gross anatomy of skeletal muscle. If we are seeing here, this is tendon. Okay, so this is tendon. The tendon is like connective tissue that anchors muscle to bone. It transmits force generated by muscle and muscle contraction to the skeleton. In tendon you can see outer part will be like epimysium. This epimysium also known as outer covering of the entire muscle, and this dense irregular connective tissue, it protects and groups the muscle into your bundles. Okay, when you are going to inside the epimysium, you can see perimysium. This perimysium is surrounds each fascicle. You can see here, these are the fascicles. This perimysium is surrounds by each fascicles, or you can tell that is a bundle of muscle fibers. It contains blood vessels. Okay, it contains blood vessels and nerves. When it is going to inside the perimysium, you can see endomysium. This endomysium, it surrounds each individual muscle fibers here. This endomysium surrounds each individual muscle fiber, or you can call muscle cell within the fascicles, and it contains some capillaries and nerve fibers. So this is the basic gross anatomy of skeletal muscle. Okay, so first in this muscle fiber, this muscle fiber also known as muscle cell, it will be like elongated. You can see here, this is, you can see this image and you can appreciate this thing. It's elongated and multinucleated cell. It can extend the entire length of the muscle. It's surrounded by a plasma membrane called sarcolemma. Okay, so this is sarcolemma. Sarcolemma also known as cell membrane or plasma membrane of the muscle cell. It conducts, this is the main, sarcolemma is important for conduction of action potential. Next, inside the sarcolemma you can see myofibril. This myofibrils is like contractile elements inside each muscle fiber. It's run in parallel along the length of the muscle fiber. It composed of repeating units called sarcomeres. Okay, this myofibrils is composed of repeating units called sarcomeres. Okay, so now we are going to see the text of the first aid guide. First one, each muscle is composed of individual muscle cells called fibers that usually run the entire length of the muscle. Okay, each muscle fiber contains hundreds of fibrils arranged in parallel. Each fibril composed of sarcomeres connected in series N to N. So sarcolemma also known as plasma membrane of the muscle cell, we already discussed. Its main function is conduction of action potential. Myofibrils are contractile structures within the muscle, and it's run in parallel along the length of the muscle fiber and composed of repeating units called sarcomeres. Next one, sarcoplasmic reticulum. The sarcoplasmic reticulum will be like intracellular structure similar to endoplasmic reticulum, and it's important for calcium storage. So this is very important for your basic physiological. They might be asked this type of questions. Okay, so in sarcomere you can see functional contractile unit a muscle. Sarcomere also known as functional contractile unit of a muscle and composed of thick. Thick means mota, means myosin, and thin means actin. Thin we know thin filament will be like actin. Thick filament myosin, thin filament actin. It arranged end to end within the myofibrils. So, this actin and myosin. What is actin? Actin means actin is a thin filament involved in contraction. The myosin will be a thick filament with heads that bind to actin to generate force. Okay. So, now we are going to see the sarcoplasmic reticulum. The sarcoplasmic reticulum is specialized endoplasmic reticulum in muscle cells. It stores and releases calcium ions for needed for contraction. Next one. Next you can see here T tubules. This T tubules, why T means transverse tubules. These transverse tubules is like invaginations of sarcolemma that penetrate deep into the muscle cell. It carry action potential from the surface into the muscle interior, closely associated with terminal cisterns of sarcoplasmic reticulum to form triads. I will explain these things while using images. Okay. Triads means made of one T tubule plus two terminal cisterns. What is cisterns of sarcoplasmic reticulum? And allow for synchronized calcium release and uniform contraction. So, this is the physiology of muscle contraction. First of all, that muscle will get signal transmission. That means action potential travels along the sarcolemma and it enters into the T tubules and it activates dihydropyridine receptors. Okay, and it will enters and it will activate dihydropyridine receptors, or you can call DHPR. Then after calcium release mechanism, we can see that is dihydropyridine receptors are voltage gated ligand type calcium channels. This interacts with the ryanodine receptors on the sarcoplasmic reticulum. Then after this ryanodine receptors are open to release calcium into cytosol. The muscle contraction, the calcium binds to troponin, exposing binding sites on actin. The myosin binds to actin power stroke contraction. This ATP is required for both contraction and relaxation. Why this thing is important? What clinically we have to know about these things means this importance of T tubules. It ensure uniform of contraction of all parts of a muscle fiber. If T tubules are damaged or missing, uncoordinated muscle contractions, if T tubule are damaged and missing, we can see like uncoordinated muscle contractions and weakness. So, guys, if you are seeing this image, you can see here this is healthy muscle cell. So, here this healthy muscle cell, in this healthy muscle cell we can see this transverse tubule. This is very important. What's the role of this transverse tubule means, T tubule means, you can see here in a healthy skeletal muscle cell the action potential travels along the sarcolemma. Then after you can see the outer membrane of the muscle fiber and quickly dips into the cell via the transverse tubules or T tubules. These are the invaginations that transmits. Here you can see this image, this action potential. Then after here its invagination to inside the invaginations that transmit the signal deep into the cell. The T tubules are closely associated with the sarcoplasmic reticulum, which stores calcium when the action potential reaches the sarcoplasmic reticulum. It triggers the uniform release of calcium ions across the all myofibrils. This even calcium release causes a synchronized contraction of the entire muscle fiber, leading to smooth and effective movements. Okay, so this is the synchronized contraction. The calcium ions are released evenly, then that muscle will get contraction. Okay, so this is the about healthy muscle cell. Next, you can see here fever transverse tubules. The fever transverse tubules means it's like pathology of uncoordinated contraction. So here now let's compare that to what happens when there are fever transverse tubules with a reduced number of T tubules. The electrical signals falls to reach all areas of a muscle fiber simultaneously. As a result, calcium is released unevenly. This leads to uncoordinated contraction, meaning parts of the muscle contract at different times, reducing strength and efficiency. This scenario is common in aging cells, aging muscle of or certain muscle disorders, highlighting how critical T tubules are for muscle function. Okay. Next one, intracellular calcium regulation. Here you can see mechanism of contraction. Let's zoom in at the molecular level. This is the triad system, the interface between the T tubules and sarcoplasmic reticulum. First one, T tubule, DHD, DHPR and RYR. That means T tubule, you can see here this is T tubule. This T tubule mostly it carries the action potential. Then after we can see here that is dihydropyridine receptor, that means L type ligand type calcium channels. It is also called dihydropyridine receptors or DHPRs are located on T tubule. Okay. So then after the T tubule membrane, then after we can say this activate the ryanodine receptors. This thing mainly it will activate ryanodine receptors. This ryanodine receptors on the sarcoplasmic reticulum. Once it activated, the ryanodine receptors will open and releasing a flood of calcium into the cytoplasm. You can see here this image. Okay. And it will release into the cytoplasm, which initiates myofibril contraction. To relax the muscle, calcium must be cleared. This is done by smooth endoplasmic reticulum. Calcium ions calcium voltage gated channel, its pump which moves calcium back into the sarcoplasmic reticulum, and additionally the exchanger helper to extrude calcium sodium calcium channel. Exchange helper helps extrude calcium from the cell and swapping it with sodium. Okay. Here what are the main important things clinically? If you are seeing here, why is this important clinically means, let me give you some few examples. First one, malignant hyperthermia. In malignant hyperthermia, we can see like mutation in ryanodine receptors causes excessive calcium release, leads to sustained muscle contraction, heat production and rigidity. So for the treatment we have to use intravenous dantrolene, which inhibits ryanodine receptors. Next one, muscle weakness in aging. Aging reduces the density of T tubules, leads to poor calcium distribution, weaker uncoordinated contractions. And next one you can see like heart failure in cardiac muscle. It's like abnormal calcium handling plays a major role. Drugs targeting like smooth endoplasmic reticulum calcium ions or calcium channels are part of therapeutic strategies. Okay. So here we can see some pathological condition, that is why this T tubules are important. It ensures uniform contraction of all parts of a muscle fiber. If T tubules are damaged or missing uncoordinated muscle contractions weakness, the ryanodine, and then after the what's the importance of clinically importance of ryanodine receptors means this ranodin receptors. Because of this ranodin receptors, we can see malignant hyperthermia. We already discussed. For treatment, we can give here. Why this malignant hyperthermia is very dangerous? Because whenever that muscle getting malignant hyperthermia, the hypothermia can cause muscle rigidity. The muscle, because of that muscle rigidity, we can see acidosis. Then after breathing related problems. Okay, so for that thing, what we have to do, we can give dantrolene. Why? The dantrolene intravenous injection can inhibit ranodin receptors and preventing calcium release. Next one, you can see Duchenne muscular dystrophy. It's like genetic absence of dystrophin, a structural protein connecting the cytoskeleton of muscle fibers to the sarcolemma. It leads to sarcolemma fragility, progressive muscle damage. How we can diagnose this thing? The elevated of creatinine kinase levels. The CK levels will increase, elevated. Genetic testing or muscle biopsy. For management, we have to give supportive therapy and corticosteroids and gene therapy based on under research. Next one, hypocalcemia and muscle excitability. Low extracellular calcium increases neuromuscular excitability, can lead to tetany, muscle spasms. Okay, so these are the main things about organization of muscle cells. Okay, so next we are going to discuss about types of skeletal muscle fibers. The muscle fibers have some types. So here skeletal muscle is made up of different types of fibers that serve different functions. They are broadly divided into two types, type 1 and type 2. So first we are going to discuss about type 1. In type 1, also known as slow or red muscle. The type 1 fibers also called slow twitch or slow oxidative fibers. Here's what you need to know as a basic things means. It's a small fiber size and small motor units. It activated for fine sustained control and low myosin ATPase activity because it contracts slowly. High mitochondrial content and oxidative enzymes, and we can say like high myoglobin. It gives it a because of this high myoglobin, it gives the red appearance. Rich capillary supply, good oxygen delivery, fatigue resistant, good for endurance. So for example, these fibers dominate in postural maintenance or postural muscles like a paraspinal muscles, which keeps us upright all day. Okay, so clinically, why this thing important means type 1 fibers use aerobic metabolism, which means they rely on oxygen and mitochondria to generate ATP. That's why they don't tire easily. Okay, next one, type 2 skeletal muscle. This type 2 muscle fiber will be like fast and white fibers. These type 2 fibers are fast twitch fibers built for power and speed. It's like large fiber size, large motor units for powerful movements, high myosin ATPase activity, and it contracts quickly. Few mitochondria, low myoglobin, rely on anaerobic glycolysis, fatigue quickly. It used in activities like sprinting, jumping, or lifting heavy weights. And the clinically importance will be like these fibers store glycogen and generate ATP via anaerobic glycolysis, making them prone to quick fatigue but excellent for brief bursts of energy. So now we are going to about muscle proprioceptors. How your body senses muscle length and tension. This is crucial for motor control. This is crucial for motor control and injury prevention. So first one, we are going to discuss about muscle spindle. The muscle spindle will be like stretch sensor The muscle spindle is a sensory receptor located within the body of the muscle. It senses muscle length and speed of stretch, innervated by type 1A and 2 sensory fibers. When a muscle is stretched, it sends signals via sensory neurons to the spinal cord. It facilitates contraction of the agonist muscle and inhibition of antagonist. So, here we have a mnemonic. How you can easily remember this thing means, whenever you are getting stretch, the spindle fibers will get contracts to resist stretch. Its main function will be like it's a negative feedback system to prevent overstretching and muscle damage. For example, knee jerk reflex, patellar reflex, tapping the patellar tendons such as the quadriceps and spindle activates reflex contractions. Okay. So, next one, Golgi tendon organ. In this Golgi tendon organ, like it's like tension sensor. It's located in the tendons, not the muscle building. Mostly it's present in tendons. This senses muscle tension innervated by type 1B sensory axons. It inhibits alpha motor neurons via inhibitory interneurons. It leads to muscle relaxation when tension is too high. So, mnemonic will be like for Golgi tendon organ, tension, tendon organ fires, tons of contraction. Okay. So, main function will be like it protects muscle and tendon from excessive force. Clinically importance will be like when you try to lift something too heavy, that Golgi tendon organ helps by inhibiting the muscle to prevent rupture. Okay, so this is the main function. So, here you can see here this is the spinal pathway. Okay, let us walk through this feedback loop using the spinal cord circuit. Here muscle spindle sends this muscle spindle sends stretch information via 1A sensory axons to dorsal root and spinal cord like this. Then after it excites by alpha motor neurons, this green one, it excites by alpha motor neurons to cause contraction of that same muscle agonist. This Golgi tendon organ via 1B sensory axons senses too much tension, activates inhibitory interneurons, inhibits alpha motor neurons, muscle relaxes. So, why is this important? In clinically we have to understand that upper motor neuron versus like lower motor neuron lesions. In upper motor neuron lesion, the hyperreflexia due to loss of inhibitory control over the muscle spindle reflexes. The lower motor neuron lesions will be like hyperreflexia or areflexia, means absence of reflex due to loss of motor neuron input. And we can differentiate with this thing spasticity versus rigidity. Spasticity mostly we can see in stroke or cerebral palsy patients is related to overactive spindle reflexes. Next one, Golgi tendon reflex testing, like it is difficult to test clinically, but its dysfunction can result in tendon tears from over exertion. Okay. So, this is the Golgi tendon, about Golgi tendon organ. So, next we are going to discuss about sarcomere. In sarcomere we can see like structure and clinical relevance, and this is the about skeletal and the cardiac muscle. Okay. So, what is a sarcomere? The sarcomere is the functional contractile unit of striated muscle found in both skeletal and cardiac muscle. It spans the distance between two Z lines, and it contains organized actin myosin and myosin thick filaments. It is the basic repeating unit that shortens during muscle contractions. So, the mnemonic will be Z to Z is the zone of contraction. You can see this image here, this Z to Z, it is a zone of contraction. Okay. So, let us break it down by band by band and the line by line about sarcomere structures. First we are going to discuss about Z line. You can see this is the Z line, you can see here, you can appreciate this image. Okay, first of all, this Z line is the like the boundary of, you can tell Z line or Z disc. It's like the boundary of each sarcomere thin actin filaments attached here and contains vimentin and despin tumor markers in muscle origin tumors like rhabdomyosarcoma. If any patient have compartmental syndrome, then after the compartmental syndrome is if it's going to severe cases, then we call that is rhabdomyosarcoma. And also anchors titin, a protein that connects myosin to the Z line, maintaining sarcomere elasticity. So this desmin is a high yield marker for soft tissue tumors involving muscles. Okay, next one, I band. You can see here, this is the I band. You can see in between, or you can call I. Why this name we got I band? I means isotropic band. It contains only actin, means thin filaments, only thin filaments. Okay, only thin filaments, that's why we call that is, it contains only actin, thin filaments. It spans across two sarcomeres on each side of the Z line. This I band means isotrophic band, gets shorter during contraction. We have a mnemonic that is I band is isolated thin filament that decreases during contraction. Next we are going to discuss about M line. This M line means myosin attaches to M line, M for myosin, simple. Okay, next we are going to discuss about A band. A band also known as anisotropic band. This A band is like anisotropic band. It contains entire myosin filament, like thick filament, including overlap with actin. A band remains the same length during contraction. The mnemonic will be A band is always the same. It will not going to change. So this is the mnemonic. You can see here, this is the A band, always the same. While contraction also it will not change, while relaxation also it's not going to change. Next one we are going to discuss about H zone or H band. This H band will be like it's located, you can see here. Okay, this H band is located in the center of the A band. This is A band, right? In center of the A band you can see H band, so that's why we call located in the center of the A band, and it contains only thick filaments like myosin with no actin overlap. It shrinks during contraction as actin slides over. Next, titin. Next we are going to discuss about titin. Titin means a huge elastic protein. It connects the myosin. Okay, it connects the myosin and to Z. It connects the myosin to the Z line. Okay, you can see here. This is titin is like one of the protein. It connects to titin to myosin, myosin to Z line. Okay, so here it provides elasticity and structural stability during contraction and relaxation. So titin gene mutation are linked to familial dilated cardiomyopathy. Okay, so this muscle contractions dynamics will be, if you are seeing this image, you can see here what happens during contraction. The actin slides over myosin, pulling the Z lines closer. The I band and H zone will get shrink. A band stays the same, and we can easily understand this topic when we are discussing about sliding filament theory. Okay, that time I am going to discuss little deep. So what's the main clinical relevance about these things means, if any patient have muscle tumors and immunohistochemistry like desmin and vimentin, It's like sarcomere associated markers used to diagnose rhabdomyosarcoma and leiomyosarcoma. The Desmin positive tumor, it's like think about muscle origin, and titin and cardiomyopathies, mutations of TTN gene, titin coding titin like familial dilated cardiomyopathy. It's like dilated cardiomyopathy, large heart with reduced function. Titin mutation is a common genetic cause. Skeletal muscle, skeletal sarcomere versus cardiac sarcomeres means we have many similarities. Both have Z lines, A bands and H zones, etc. Like contraction mechanism is the same with sliding filament theory. The difference will be cardiac muscle cells have intercalated discs and that are connected via gap junctions. The skeletal muscle is voluntary, cardiac is involuntary. Here in your exam they might be asked this type of questions: which sarcomere band remains the same length during contraction? That means A band, because A band is not going to change while contraction, while relaxation. Next one, which band disappears completely during full contraction? That is H band, because the H band, if actin filaments fully overlap myosin. Next one, a tumor positive for Desmin likely originates from which tissue type? That is muscle. Which protein connects myosin to the Z line and maintains elasticity? That is titin. Okay, so next we are going to see the main important topic and very little hard topic, that is the neuron action potential. Okay, so what is an action potential? The action potential is a rapid temporary change in the electrical potential across the membrane of excitable cells like neurons and muscles. It is the fundamental mechanisms for nerve impulse transmission, the muscle contraction. It's like driven by ion movements across the membrane, especially sodium and potassium. So you can see this graph. You can see here the action potential thing. You can see here, this is the components of the action potential. Here you can see the resting membrane. Resting membrane potential will be like a minus 70 millivolts, and you can see threshold potential, that is minus 55, and we can see depolarization, this thing, and we can see overshoot, this one. Next one, repolarization is coming down, that is repolarization. Next, hyperpolarization. Next one, return to resting membrane state. Okay, so first one, what is resting membrane potential? Minus 70. What is important means this here, this resting membrane potential will be like it maintained by high potassium permeability. You can see this image. This is resting membrane potential. This resting membrane potential will be like, you can see high potassium permeability through leak channels, low sodium permeability. Inside of the membrane is more negative than the outside. The sodium potassium ATPase pumps, like 3 sodium out it will go, and 2 potassium will go inside for maintaining gradient. So clinically importance will be like for resting membrane potential is clinical importance will be digitalis, and avabatin inhibits sodium potassium ATPase affects the resting potential. Next one, threshold. Next we are going to discuss about threshold, what it means. Threshold potential is usually like around minus 55 to minus 55 millivolts. You can see this image. If a stimulus depolarizes the cells to this point, action potential is triggered, all or none law. Like mnemonic will be like threshold means trigger zone. Okay. Next one, depolarization. You can see this image. In depolarization means from threshold it's going to increase straight and it stopped till plus 30. So we call that is depolarization. In this depolarization, we can call we can also known as upstroke. Here, like this is voltage gated sodium channels open, and massive influx of sodium makes the membrane potential more positive. The membrane potential can reach plus 30 to plus 35 millimoles. This is the overshoot phase. Mostly we can see this thing in lidocaine, like lidocaine means a local anesthetic. It blocks sodium channels, it prevents the depolarization and no pain conduction. Next one, repolarization. In this repolarization, we can say downstroke. The sodium channels will close and inactivates. The voltage gated potassium channels will open and potassium efflux, we can see here, and brings the membrane potential back down towards the negative. So here main thing is the potassium permeability is now exceeds even resting state, preparing for next step. It's preparing for next step. Next one, hyperpolarization. In hyperpolarization means after potential, like potassium channels are slow to close, so extra potassium leaks out. The membrane becomes more negative than the minus 70 millivolts up to minus 85 millivolts. Eventually the potassium channels will be closed, but to resting potential via leak channels. The mnemonic will be like hyperpolarization, or you can talk HeLa negative. So the membrane permeability changes, you can see here, that is the sodium permeability spikes during depolarization, the potassium permeability spikes during repolarization and hyperpolarization. At rest, potassium dominates via leak channels. Okay, so clinically it's important when using local anesthesia during like lidocaine or bupivacaine, which blocks the voltage gated sodium channels. It prevents depolarization, thus block nerve conduction. It used in nerve blocks and dental procedures. Next one, hyperkalemia and hypokalemia patients. In hyperkalemia, it raises resting potential closer to threshold, increased excitability initially, then sodium channels inactivation like muscle weakness. Next one, hypokalemia, it makes resting potential more negative, harder to reach threshold, fatigue, muscle cramps, and we can see demyelinated disorders like multiple sclerosis. Myelin speeds up conduction via saltatory conduction. Demyelination slowed or blocked conduction. The channel and channelopathies, we can see this is like genetic mutations affecting sodium, potassium or calcium channels can cause like epilepsy, periodic paralysis and cardiac arrhythmias. Okay, so next we are going to discuss about, this is like last topic we are going to cover in this video, that is refractory, absolute refractory period, or you can tell functional refractory period. So absolute refractory period means, the absolute refractory period means is the time during which a second action potential cannot be initiated, and no matter how strong that stimulus. It begins once the membrane reaches threshold and continues until almost the end of repolarization. It caused by inactivation of voltage gated sodium channels. Why no action potential can occur? The sodium channels are either already open or inactivated, so new depolarization is impossible. So we have a mnemonic for this ARP, means absolute refractory period. The absolute means absolutely no chance of second action potential. You can see this image here. This is the absolute refractory period. Okay, so here you can see this the classic action potential, the curve, and say the absolute refractory period zone from threshold to mid repolarization. Okay, Next, here one important impact will be is there, that is drugs that prolong sodium channel inactivation or slow repolarization while increase the duration of the absolute refractory period. In example, like you can talk about class 1 anti-arrhythmic drugs. It blocks sodium channels, prolong absolute refractory period. It used in ventricular tachyarrhythmias. Okay. After hyperpolarization means undershot, we can see like also called undershoot. It occurs after repolarization due to slow closure of potassium channels, and the membrane potential becomes more negative than minus 70 millivolts to minus 85 millivolts. The clinically important will be like the part of relative refractory period, a stronger stimulus can initiate another action potential. Okay. So, next we are going to discuss about factors determining the velocity of the action potential. The action potential factors, we can see amplitude. The larger the amplitude, is like stronger electrical force, faster conduction, rate of depolarization, and its faster sodium influx, faster propagation of action potential. Okay. So, here we can see neuron factors. In neuron factors, we can select neuron specific factors like axon diameter and myelination. The axon diameter will be like larger diameter, lower internal resistance, faster current, and myelination it's like myelin acts as an insulator, increasing membrane resistance and action potential speed. The action potential jumps from node to node via saltatory conduction, and its nodes of Ranvier conduction voltage gated channels. We have a mnemonic for this thing for fast conduction, that is big and insulated wires are fast. Okay. So, clinical application will be like demyelinated neuropathies. In demyelinated neuropathies, you can see like Guillain-Barré syndrome and multiple sclerosis. It is like loss of myelin and current leaks or slower or blocked conduction, and it affects velocity but not necessarily amplitude. In axonal neuropathy, you can see like damage to axon, fewer fibers to transmit action potential, and it causes reduced signal amplitude in nerve conduction studies. So, if you are comparing this demyelinating neuropathies and axonal neuropathies, here we have for neuropathies we have one thing that is we called conduction velocity and signal amplitude. In demyelinating, you can see the conduction velocity will decrease, the signal amplitude will be like normal and slightly reduced. In axonal, we can say like the conduction velocity will be like normal and slightly reduced, the signal amplitude will be reduced. The nerve conduction studies, you can see like non-invasive diagnostic test. It measures like amplitude, latency, delay, and velocity. Okay, and you can use this thing differentiate between like axonal and versus demyelinated disorders. Here you can see one toxin that is we called tetrodotoxin. This tetrodotoxin is neurotoxin from pufferfish. Sorry. It's like pufferfish will be like poisoning. It's caused by tetrodotoxin. It blocks voltage gated sodium channels in neurons and heart. It prevents depolarization, causing like muscle paralysis, respiratory failure, and no action potential conductions. Okay. This thing is very important for your USMLE and FMGE because they might be asked questions from this thing. Okay. So, mostly we covered everything. So, in our next video we are going to discuss about neuromuscular junctions and some clinical significance also. Okay. So, in this video what are the things we discussed? First of all, we discussed about organization of muscle. Okay, here I am just giving recap. The muscles are made of fascicles, which are bundles of muscle fibers. Okay, it's the key points. Each fiber contains myofibrils, which are made of sarcomeres. T tubules and sarcoplasmic reticulum coordinated calcium release for contraction. Dihydropyridine and ryanodine receptors play a key role in calcium mediated contraction. Clinical conditions we can say like include malignant hyperthermia, neuromuscular dystrophy and hypocalcemia and related to tetany. Okay, so T tubules allow action potential to reach all parts of the muscle fiber. The calcium release from sarcoplasmic reticulum is essential for muscle contraction. Proper reuptake and extrusion of calcium ensures relaxation. Defects in this system can lead to uncoordinated movement or dangerous conditions like malignant hyperthermia. Okay, so next you can see here we have some, if any patient have like if you are trying to management of perspective on muscle fibers like types helps to guide rehabilitation and sports training, that time you have to know two things: endurance training and resistance training. In endurance training that it increases type 1 fibers and mitochondrial content. In resistance training you can see you can increase the type 2 fiber size, hypertrophy and strength. In diseases like muscular dystrophy you may see fiber type grouping due to denervation and reinnervation. Okay, so next one, the final thing will be which sarcomere band remains the same length during contraction. I am just asking 4 questions, you can give me answers in comment section also, no problem. Which band disappears completely during full contraction? A tumor positive for desmin likely originates from which tissue type? Which protein connects myosin to the Z line and maintains elasticity? These are the 4 questions I asked. So if you know the, we discussed in this video, if you know the answers, you can write these things in comment section. Okay, next questions, and maybe they can ask in your exams or in like in while discussing about skeletal muscle system, that time they can ask these questions in ASCII and Viva and these things. So that's why I'm highlighting main topics. Based on main topics I made some question, so I'm going to ask. Okay, what causes the absolute refractory period? Answer will be inactivation of voltage gated sodium channels. How does myelin affect conduction? It increases speed via saltatory conduction. We will discuss this thing, what is saltatory conduction, in our next video. What happens in demyelinating neuropathy? In demyelinating neuropathy, the conduction velocity will reduce a normal amplitude. Next question, tetrodotoxin blocks which channel? The voltage gated sodium channel. So if you are, this video is helpful for you, so still now you didn't subscribe my channel, like, share and subscribe. If you have any doubts you can ask me in comment section. So we will meet in our next video. Till the time, take care, bye bye.